Thank you, Dror, and good afternoon. My charge over the next few minutes is to describe the evolving treatment landscape of advanced prostate cancer. And let me begin by making the case that the current treatment landscape of prostate cancer has already been transformed. And I'll present several ways of making this case. First, this is the timeline for FDA approvals in castration-resistant disease. The approvals shown above the horizontal axis are drugs that increase overall survival, and those below are drugs that have been approved based on intermediate endpoints, usually reflecting patient symptoms. The first drug approved based on overall survival was docetaxel in 2004. That led to this really kind of artificial construct of pre- and post-docetaxel in patients with metastatic castration-resistant disease to account for that only drug that increased survival. And now if you just focus on the latter part of that timeline, you see this really remarkable progress in recent years with 5 additional drugs approved based on increased overall survival. Now, it's important to note that these drugs, for the most part, were developed in parallel or at the same time, so they really didn't account for the others in their development. And this has led to lots of challenges in understanding optimal sequences and combinations of drugs. So remarkable both in just the absolute number of drugs approved in recent years based on increased overall survival. The other remarkable thing is that these drugs involve multiple different mechanisms of action. This is just a summary slide looking at the different mechanisms of action of the recently approved and emerging therapies. We have an immunotherapy, Sipuleucel-T, approved based on increased overall survival. 2 chemotherapy drugs, docetaxel and Cabozantinib, both increase overall survival. Abiraterone acetate and enzalutamide, targeting AR signaling and ARN-509, which I'll talk more about. Cabozantinib in development, targeting MET and VEGF receptor. And then a number of bone-targeted drugs, 2 drugs that target the osteoclast and prevent or delay skeletal-related events, and radium-223, an alpha particle that has been shown to increase survival and reduce the risk of symptomatic skeletal events. These drugs also impact different parts of the timeline of the prostate cancer continuum. This is the hypothetical prostate cancer continuum for a man diagnosed early with fatal prostate cancer. So that type of patient would have surgery or radiation therapy. Upon failure with the rising PSA, he goes on androgen deprivation therapy, and sometime later progresses to castration-resistant disease, subsequently to symptomatic disease, and develops fatal complications. The drugs are approved in different parts of that continuum. Docetaxel approved in 2004. Several drugs approved in the so-called post-docetaxel space, abiraterone, Cabozantinib, enzalutamide, and radium-223. And 3 drugs approved in the pre-chemotherapy space, abiraterone, radium-223, and Sipuleucel-T. And in a press release this week, we learned that enzalutamide also improves overall survival in that setting, and we expect the expanded approval of that drug in coming months. So lots of treatment options, lots of uncertainty about optimal sequencing and combination. To make the future even more complicated, there are a lot of drugs in development. And this is, by all means, simply a partial list of the drugs in phase 2 or 3 development. I've grouped them by different targets: anti-androgens, CYP17A inhibitors, a drug that targets both CYP17A and AR signaling, immunotherapy approaches, novel targets, and tyrosine kinase inhibitors. And focus for just the last minute on this last column, rather remarkable series of acronyms, very hopeful acronyms: COMET, READY, Affinity, Prospect, Spartan. So we hope the acronyms match the clinical trial results. Now, to go through all of these drugs in development would be well beyond the scope of time this afternoon. So I'm going to focus on 2 of these drugs for very specific reasons. So ARN-509 is a novel anti-androgen. The approach in its development is to move earlier in the course of the disease. And I'll talk about Cabozantinib, a very different approach with novel targets and being developed more in advanced disease, fitting its expected risk-benefit profile. So here's the places we're talking about. Cabozantinib initially being developed in more advanced disease, and then this newer approach of moving a highly active anti-androgen somewhat earlier in the course of the disease. Cabozantinib is an orally available novel receptor tyrosine kinase. It has a number of main targets, including MET, VEGF receptor, and RET. Cabozantinib is approved to treat medullary thyroid cancer. Its approval was based on the results of the EXAM trial, a randomized controlled trial in patients with locally advanced or metastatic medullary thyroid cancer. Dramatic impact on progression-free survival, difference of median of 4 months in placebo versus 11.2 months in favor of cabozantinib. So very marked separation of the curves and led to the approval of cabozantinib for medullary thyroid cancer. The biology behind this is very strong, very rational drug development, but it's worth noting that cabozantinib is a drug that there's a solid preclinical rationale for development in other cancers, including metastatic prostate cancer. And here's the evidence. MET is highly expressed in bone and bone metastases. Osteoclasts and osteoblasts express MET and VEGF receptors and respond to their corresponding ligands, HGF and VEGF. And HGF and VEGF direct crosstalk between tumor cells, osteoblasts, and osteoclasts. So targeting MET and VEGF receptor would be predicted to have an impact on bone, on tumor, and tumor-bone interactions. Cabozantinib has been evaluated in a variety of different malignancies, and it was sort of efficiently evaluated in a spectrum of malignancies in this randomized discontinuation study that looked at 9 different disease cohorts, including prostate cancer. And there was a very early, remarkable signal in that trial showing a high level of activity based on changes, post-treatment changes in technetium bone scans. So these are some of the representative images from that randomized discontinuation trial, baseline in week 12 paired images for individual patients, showing a marked improvement in uptake in technetium bone scans, as in some cases, as in, for example, this patient 2, a near complete resolution of disease on technetium bone scan. This is quite candidly a finding that had been rarely seen with any other drug, seen at a high rate in this randomized discontinuation trial that led to a lot of excitement about the drug's further development in prostate cancer. We first took the approach of expanding this trial into a non-randomized expansion cohort, several different aspects relative to the randomized discontinuation study. They were a more advanced group of patients and were treated with uninterrupted cabozantinib. So these are the patients we accrued to the so-called NRE. All patients had bone metastases, 100%. About a third had visceral metastases. Very advanced patient population with a lot of pain, symptoms related to disease. All the patients had disease progression despite prior docetaxel, and many of them had also disease progression following abiraterone, enzalutamide, and other drugs like cabazitaxel or radium-223. More than half of the patients were on concurrent bone-sparing agents with bisphosphonates or denosumab. The majority of patients had progressed, all the patients, had progressed within 6 months of prior docetaxel, and fitting with this high burden of disease, very high PSA, 194 at median at baseline, very high CT counts of 49 at study entry. A high rate of activity was observed by a number of metrics. The pre-specified primary endpoint in this trial was the post-treatment change in bone scan, so-called bone scan response. This was done by quantitative imaging, and this is a waterfall plot representing the individual patient changes in post-treatment bone scans. More than 2/3 of the patients had improvements in bone scans. More than 80% of patients improvements in bone scan. About 2/3 of the patients met the pre-specified response criteria of a greater than 30% in bone scan lesion area. As you can see, very few patients had worsening of bone scan as the best response. Each of the individual patient data is color-coded to represent prior therapy, and you see this activity observed really in all the patient categories, include those not only that had prior docetaxel, but those had additional salvage therapies, including abiraterone and or MDV 3100, suggesting that the drug has activity in highly refractory patients and that the activity of cabozantinib is not cross-resistant with other established therapies. Most of the patients had improvements in sites of measurable soft tissue disease. Here, about 2/3 of patients had improvement in measurable disease, although relatively few patients met criteria for resist response. PSA was often discordant with other metrics of anti-tumor activity. There were some PSA declines, and those patients fairly uniformly had improvements in bone scan and measurable disease, although some patients with improvements in bone scans as well as measurable disease had PSA rises. So we don't believe PSA is a meaningful biomarker for assessment of activity of this agent. This is change in circulating tumor cells. This is best response at week 12 among the patients who had more than 5 CTCs at baseline. A high proportion of patients had decline in CTCs. 92% demonstrated a best CTC decrease of greater than 30%, and a conversion rate from unfavorable to favorable about 40%, which is quite comparable with our most active currently available therapies in this disease state. In addition to objective measures of tumor assessment, there is subjective improvements in pain. The majority of patients with pain at baseline had improvements in pain. About 56% decreased narcotic use, and nearly a third discontinued narcotics entirely. As predicted from the preclinical data, cabozantinib resulted in improvements in bone biomarkers. On the left is the waterfall plot for CTX, a marker of osteoclast activity, and on the right is BSAP, a marker of osteoblast activity. And you can see that the majority of patients had reductions in both CTX and BSAP, including patients who were on concurrent treatment with the bisphosphonate. So in summary, cabozantinib has clinical activity in men with treatment-refractory metastatic CRPC, high rate of bone scan responses, high rate of progression of measurable disease, marked improvement in pain in many patients, and substantial reductions in CTCs. In data I won't present today, cabozantinib also has activity at lower dose, and at the doses studied has a safety profile similar to other receptor tyrosine kinase inhibitors. And on the basis of this phase 2 data, there are now 2 ongoing phase 3 studies that will evaluate the effects of cabozantinib on overall survival, the COMET-1 trial, and pain, the COMET-2 trial. So here's the design of the COMET-1 study, about 1,000 patients with metastatic castration-resistant prostate cancer, bone metastases, and progression despite prior docetaxel and abiraterone or enzalutamide will be randomized 2-to-1 to cabozantinib versus prednisone with a primary endpoint of overall survival. That study recently completed accrual, and we expect the top-line results sometime in 2014. COMET-2 is a smaller trial with the primary outcome of pain, randomization to mitoxantrone prednisone, a drug combination that already has an FDA-approved label for palliation of pain in this setting. So we've talked about a novel targeted therapy in particularly advanced disease, highly refractory patients. I'm not going to talk about a more validated target of the androgen receptor looking at ARN-509 in earlier stage disease. So there's quite a large body of evidence to support the conclusion that AR overexpression is a central mechanism for castration-resistant prostate cancer, certainly not the only mechanism, but most tumor cells show increased AR mRNA and protein levels. Some show gene amplification. About 10% of cells have gain-of-function AR mutation, and AR overexpression leads to resistance to anti-androgens in preclinical models. And notably, bicalutamide in that setting exhibits agonist activity in the setting of AR overexpression. ARN-509 is a next-generation anti-androgen. It binds to the ligand-binding domain of the androgen receptor. It blocks nuclear translocation of AR and prevents binding of AR to the androgen response elements on DNA. Very potent activity in preclinical models. This is a mouse model of castration-resistant prostate cancer showing a really marked dose-dependent regression in tumors with ARN-509, substantially greater efficacy than with older anti-androgens. In contrast to bicalutamide, ARN-509 does not exhibit AR agonist activity. See that at higher doses, bicalutamide results in agonist activity in these preclinical models. In contrast, ARN-509 at all studied doses results in tumor regressions. ARN-509 has now been studied in a completed phase 1 trial, recently published by Dana Rathkoff at Memorial Sloan Kettering. This is the design of the phase 1 study. Important and interesting sort of novel aspect to this study was the incorporation of DHT PET scans to demonstrate in vivo target engagement, and we believe that's been very useful in selection of optimal dose, as I'll describe in a moment. So from that phase 1 study, really clear evidence of efficacy. That's a waterfall plot shown on the right, showing the majority of patients had a PSA decline, with most patients achieving a greater than 50% confirmed decline in PSA. There was activity seen at daily doses ranging from 30 to 480 milligrams. The PK exposure was dose proportional, and quantitative imaging, and I'll show you some examples of that in a moment, demonstrated robust targeted engagement of greater than 90%. The MTD was not reached in the phase 1 trial. There were no seizures specifically, no grade 3 or 4 fatigue, and the most common AEs included fatigue, back pain, dyspnea, and GI symptoms. So here's the DHT PET scans that I promised, and then you see response by more conventional criteria. So you can see this high-level target engagement with following treatment with ARN-509 with corresponding regression in this iliac crest bone lesion. This is an example from a single patient. So here's the data summarized a different way, providing rationale for the dose selection of 240 milligrams daily. This oval shape here represents the area with high-level target engagement by DHT PET. 240 falls right in the middle of that spectrum. And then these are the steady-state plasma troughs from the phase 1 study. Here's 240, and you can see it's right in the range that we would like to be in based on the preclinical suggestion of the maximally effective dose based on serum concentrations. So those results inform the design of the phase 2 portion of the trial. And in this study, 3 different disease cohorts have been evaluated. Men with non-metastatic castration-resistant prostate cancer, metastatic CRPC with no prior salvage therapy, and then metastatic CRPC following disease progression on abiraterone. Here's really just the top-line results from that. Very high response proportion, all but one patient with a PSA response in the non-metastatic high-risk CRPC. Similar, very impressive waterfall plot in the treatment-naive metastatic CRPC. And you see some responses, but a substantial fall-off in activity in the abiraterone pre-treated patients. So this is a sort of a consistent theme we're seeing with the new highly active anti-androgens, suggesting cross-resistance with CYP17A inhibitors, and that's going to be the subject of a lot of future investigation. But bottom line, these waterfall plots, as well as this waterfall plot, consistent with really the best activity that's been seen with AR-targeted therapy. Now, so in summary, ARN-509 is active in men with castration-resistant disease. There's robust PSA responses at week 12 for patients with non-metastatic CRPC as well as treatment-naive metastatic disease. About a quarter of patients had responses in the post-ABI setting. ARN-509 at 240 milligrams a day has a manageable safety profile. Most common AEs were fatigue and GI disturbances. No serious adverse events, no seizures, no grade 3 or 4 fatigue. And we believe that these data support further development of ARN-509. So how does one do that in what is now a really complicated treatment landscape? And I think you do that with several randomized controlled trials, but the first phase 3 trial of ARN-509 is the Spartan trial. This will look at men with non-metastatic castration-resistant disease, so disease progression despite standard androgen deprivation therapy for men who have not yet developed metastatic disease and who have particularly high-risk features, in this case defined as a PSA doubling time of less than 10 months. They'll be randomized to ARN-509 plus continued androgen deprivation therapy versus placebo plus ADT, with the primary endpoint of metastasis-free survival, key secondary endpoint of overall survival. So this is really looking at the idea of metastasis-free survival as an approvable intermediate endpoint with supportive data from overall survival. And there will be additional phase 3 studies planned of the same compound. And with that, I'll conclude, and thank you for your attention.